Hi everyone. In this experiment, you're going to learn about the effects of cleaning products on microbial growth. And the summary of this experiment is you're going to take two agar plates and you're going to streak something before cleaning it and after cleaning it, see if it does have any difference to clean something or disinfect it. So the material you're going to need to do this experiment is you're going to need two agar plates. You're going to need a Sharpie to label your agar plates, the little sterile water that came in your kit. You need a cotton swab and 2 inoculating loops per plate. So you need 2 cotton swabs and 4 inoculating plates. And I'm going to show you how we're going to do this. So I want everyone to choose something at home that they want to streak on these plates that you touch often. I chose my car keys because it's something I touch often. I don't want you to choose something like the top of a window that you never touch, something that's touched. You all can do phones if you want. You can do laptops, you can do iPads, you can do doorknobs. That's a good one. So that's something that's touched often by your hands. And then we're going to clean it. You can even do a table if you feel like you're always using that table. So the first thing I'm going to do is I'm going to label my agar plates and then we'll talk about the cleaning products. So as always, when we label agar plates, there's the lid and the agar. Don't open them. I'm just trying to demonstrate again that we always write on the agar side because you can use the lid, and you always write on the sides of the plate just so that you can see the bacterial colonies later. So I'm going to start by labeling my plate like always. I'm going to start with my name, the date, and then I'm going to write here keys, no cleaning. Okay, then I'm going to take my other agar plate, and again, I'm going to write my name, the date, and then I'm going to put keys. And I'm going to clean my keys with Clorox wipes. We'll talk about the things you can use in a second. So let me write that. Keys, Clorox wipes. So just a label to know what's clean and what's not. Now, for this streaking, we're going to do streak plate technique, which you all learned so far. People who are experienced don't have to label quadrants like we did in the streak plate technique demo. But since you're still learning, I'm going to draw this again. So what I'm going to do with my plate is I'm going to split it in half and write 1 at the top, and then split the bottom into 2 quadrants labeled 2 and 3, like that. Same with my other plate. Okay, 1, 2, 3. So both top quadrant and remember the goal of streak plate technique was to dilute the culture so that you end up seeing individual colonies. Okay, to start off, the things that you're going to need is choose something you touch often. Again, you can do something like your phone, you can do keys. I'm going to do keys because I did my phone before. So here's my keys, they're touched all the time. I don't think they've ever been cleaned before this experiment, so this is something good to test. And now we're going to talk about cleaning products quickly. So cleaning products that I found in our home that we use common is Clorox wipes. So this is what I'm going to use. I just want to do other examples. We have rubbing alcohol and we have hydrogen peroxide. I want to mention, we're going to learn this later, that we do not use hydrogen peroxide for cuts or anything. It's much better to use soap and water. You'll learn about why that is later, but I don't know why we have it, but we do have hydrogen peroxide. It's a full bottle. We have this method stuff that's from Target that we use a lot to clean counters. And then hand sanitizer. I thought hand sanitizer was a good one because when we travel, I always take hand sanitizer with me and wipe my phone and wipe everything. I don't know how effective it is, so maybe one of you wants to test like wiping your phone before and after with hand sanitizer. So those are the material. Now, I'm going to do Clorox wipes, and Clorox wipes or Lysol wipes, whatever wipes you're using, look at the container. You can also use vinegar. Some of you clean your house with vinegar. So it says on it that it kills 99.9% of bacteria and viruses. That is a huge statement to make. And so when a company makes a statement like this, you've actually had to test it. The Clorox company did not test 99.9% of bacteria and viruses in the world. But if you go to the back to the little subscripts that no one ever reads, you'll see they list out this claim that it kills 99.9% of those microbes. They've tested human coronavirus, influenza A2 virus. They've tested various gram-negative and gram-positive bacteria. So they have Klebsiella pneumoniae, Staphylococcus aureus, methicillin-resistant Staph aureus. So various things that they've tested. So this is a good job if you want to be a microbiologist for the Clorox company. They're probably doing things similar to this. Okay, we're gonna start the experiment. So I'm going to take my keys that I touch all the time, and you're gonna start by taking your sterile cotton swab and opening it. And so you wanna get your sterile cotton swab, and here I have my sterile cotton swab, and you are going to put it in your little sterile water. You're gonna wet the Q-tip or cotton side of it. And the reason I'm wetting it is just it's easier to grab bacteria or potential microbes off of dry surfaces once you wet them. And then I'm just going to streak everywhere. Okay, streaking my car keys, just basically trying to get as much as I can. I'm gonna put my keys down, and I'm gonna go to the plate that says keys no cleaning or not clean. I'm gonna grab it. There's my agar, and remember with the streak plate technique, I'm gonna do quadrant 1 all the way to the middle. Stop, and then you can dump this cotton swab, close the plate. And now what we're going to do is you're going to open an inoculating glue, because that's how a streak plate technique is done. I open it, go to your plate, grab a little of your inoculum from quadrant 1 into quadrant 2. There's that, close it. Open a new inoculating loop, and then go from quadrant 2 to quadrant 3. That's how it's done. I'm gonna close it. Okay, that's ready to be incubated. So I'm going to set that there. And then I'm going to do my next plate, the after clean. So here are my keys. I decided to do Clorox wipes. If you do hand sanitizer, you can just put hand sanitizer directly on the surface or on a tissue or something, just how you actually do clean at home to test it. Okay, I'm cleaning them. By the way, I want you to read the directions on here that I don't think people do. It says on here, to disinfect and deodorize hard non-porous surfaces, wipe the surface enough for the surface to be visibly wet, and then wait 4 minutes. So I'm wiping them, and my keys look visibly wet. I can see the liquid spots on it, and you're supposed to wait for 4 minutes. And their claim is basically that after 4 minutes, that's when the microbes have died. I anticipate that when they did this in their lab, they probably tried different time points and then streaked various plates, if I was to take a guess, to see the microbial growth. Okay, we're gonna assume that 4 minutes have passed. So now I'm going to take another sterile cotton swab, and remember with these experiments you've already washed your hands. Again, dip it in my sterile water, go like this to try to get as much potential bacteria as possible. Put my keys down, do quadrant 1. Okay, that goes in the trash. Open another new inoculating loop, and then go from quadrant 1 to quadrant 2. That gets dumped. Open another new inoculating loop and go from quadrant 2 to quadrant 3. Okay, my plates are both done. So we always incubate agar side up. And since this experiment is being done at home, we are going to incubate at room temperature. I anticipate for about 48 hours, 2 to 3 days, and we'll see what happens with our plates before and after cleaning, and if these do indeed kill 99.9% of bacteria and viruses.